Hi, I will be discussing the paper SARS-CoV-2 infects cells after viral entry via clathrin-mediated endocytosis. It was released earlier this year. This paper identifies that SARS-CoV-2 enters and infects cells rapidly using clathrin-mediated endocytosis. As the current pandemic has been proven to be the greatest public health crisis of the century, creating therapeutics is key in the fight against COVID-19 and future viral infections. Understanding the mechanism of how these viruses enter a cell is also key, as targeting early steps in the viral infection has been shown to yield high-efficacy therapeutics. SARS-CoV-2, known as Severe Acute Respiratory Syndrome Coronavirus 2, causes COVID-19. The SARS-CoV-2 has an RNA genome which encodes the viral proteins it will need to replicate in the host cell. The viral molecule has a spike glycoprotein, which has S1 and S2 domains as seen on the figure in the slide. The S1 subdomain is the receptor binding domain, which will bind to the host receptor, which is the angiotensin converting enzyme 2, or ACE2. The ACE2 receptor is commonly found in lung epithelial cells. The S2 domain is used for viral and cell membrane fusion. So in this paper, the researchers looked to confirm and quantify their prediction that SARS-CoV-2 uses clathrin-mediated endocytosis to enter and infect the host cell. They wanted to know where the viral and cell membrane fuse by asking whether endocytosis is clathrin-dependent or independent. They hypothesized that SARS-CoV-2 undergoes a similar endocytic mechanism to SARS-CoV or the MERS-CoV, which were endocytic too. They used HEC293 cells and purified spike protein, and there was DAPI-stained nucleus in the study. Based on their findings, this is the general mechanism of clathrin-mediated endocytosis for viral infection by SARS-CoV-2, which is the figure on this slide. Proving that viral entry occurs in a clathrin-dependent manner allows for the development of therapeutics targeting either the clathrin coat or even the endosome formed after coat removal. The viral particle enters the cell in an endocytic manner and is covered with the clathrin protein coat until it becomes an early endosome, after which viral replication occurs and then exocytosis. Clathrin has 3 heavy chains and 3 light chains, which are absolutely key to its function. This clathrin-dependent endosomal mechanism provides the virus with increased survivability in the host, as the clathrin coat prevents viral structures from detection from the host cell immune system. So, firstly, to test for endocytosis, they did an endocytosis assay using purified spike protein, and the imaging was done using confocal microscopy. So as seen in figure 2, they used HEC293 T cells, which were either wild type or overexpressing ACE2. The cells were incubated and then given an acid wash to remove the surface spike protein, and therefore, as seen in the figure 2A, that spike protein is internalized via ACE2, and you can see this as you can only see the spike protein and ACE2 in the HEC293 cells, which were expressing ACE2 and not the wild type ones. And only HEC293 ACE2 cells show the spike fluorescence in the B of figure 2. Additionally, on figure 3, to prove that there was actually internalization, you can see that at the 5-minute interval, you can already see spike protein, which is a key indicator of endocytosis. So, to test whether there was clathrin-mediated endocytosis specifically being used by the SARS-CoV-2, they decided to examine the internalization by using in the presence of 2 drugs. One is dinosaur, which it means like once there's dinosaur in the system, there's no more fission of the clathrin-coated pits, meaning no clathrin-mediated endocytosis. And then on the other one was pitstop, where the clathrin heavy chain can no longer interact with the adapter protein needed to make the clathrin coating. And you can see in the figure that with the addition of both drugs, it reduces SARS-CoV-2's endocytosis, and this suggests that there's clathrin-mediated endocytosis. As a note, they used a controlled DMSO and a negative pitstop 2 negative control to validate the results. Now, to actually validate that clathrin is very key to this clathrin-mediated endocytosis in the context of SARS-CoV-2, they did a clathrin heavy chain knockdown, in which they had a predefined siRNA pool, silencing RNA, and they used that to knock down the clathrin heavy chain to determine the role of the clathrin. And as you can see, HEC293 T cells with ACE2 saw 70% less clathrin heavy chain levels. Knockdown led to less spike protein internalization, which is seen on the circled portion of the figure. Additionally, they redid this test in Vero monkey kidney epithelial cells and A549 cells, which are also models for lung epithelial cells, and found the same results. So overall, the researchers demonstrated that there's clathrin-mediated endocytosis occurring and that it's key to the infectivity and internalization of SARS-CoV-2. So malaria drugs like chloroquine block clathrin-mediated endocytosis by depleting the clathrin heavy chain, which then blocks the process, not allowing for viral entry in theory, which is a potential application of such research. The researchers do not propose any mechanisms or potential therapeutics which could be developed now that the viral entrance mechanism is known, which is something future researchers must consider. The researchers found that viral RNA enters to the cytosol from the lumen of the endosome in clathrin-mediated endocytosis. This research confirms that SARS-CoV-2 uses this clathrin-mediated endocytosis, which is important as this can be used to target the ACE2 spike protein interaction or the clathrin coating or even the early endosome that forms. They also state that previous studies in just SARS-CoV have shown conflicting views on clathrin dependence or independence, which leads to questions in this research. Although this research confirms clathrin-mediated endocytosis, drugs targeting clathrin-mediated endocytosis are not the only ways to fight COVID-19, and vaccine development, a large means of preventing further spread and mortality, also requires a deep understanding of virus entrance mechanisms, further pointing to the importance of the research. Thanks for listening.